All right, so we've talked about how to copy the DNA. We've talked about how to use the DNA. Now we need to talk about how to change the DNA. So there's a variety of ways you can do this, and they basically fall into two camps. You can mutate it by making a mistake in the code, or you can do a variety of other things to share it with your friends. So we're going to talk about all of those, but first we need to understand this. What is a mutation? If you ask most people on the street, they're going to come up with some movie-influenced, Hollywood-influenced idea about what a mutant is. We want to understand what it is from a biologist's perspective, which means that it's a change in the code of the DNA. What is the consequence of a change in the code of the DNA? Potentially change in the protein. Not always, right? We do know that there are mistakes you can make in the DNA that don't have any consequence. I'll give you two examples. One kind of mistake would be, since our code is degenerate, there's more than one way to code for leucine. So if you make a change in the three-letter sequence that still means the same thing, no effect. Or there are sequences of amino acids that will form the same structure, even though they're made of different amino acids. So you could switch out a leucine for an alanine, and really the structure would not be changed. Okay? Or you could make a change in the regulatory non-coding part of the DNA, which is a little more forgiving, and maybe that change doesn't do anything either. But maybe it does, okay? So it can result in a change in the protein. Now, mutations may be neutral, right? They don't have any effect. They might be good, or they might be harmful. Mutagens are things that cause an increase in the rate of mutation. The mutagen is blind. It doesn't know whether or not the change that's being made is going to help you or hurt you or not do anything. Mutations can occur spontaneously without a mutagen. How would that happen? What is the most likely source of those kinds of mutations? If you had a book and you wanted to make a copy of it and you were doing it by hand, what is the most likely way that you could introduce mistakes into your copy? By copying it at 2 o'clock in the morning when you're sleepy and making typos, okay? And so when we copy the DNA, every time we copy the DNA with a DNA polymerase during replication, we could potentially introduce a mistake. DNA polymerase is really, really good at copying, and it has proofreading activity. So if it makes a mistake, it'll frequently fix it, but not always. 1 in about 1 million base pairs gets accidentally put in wrong. All right, so we understand what mutations are. Now for microbes, mutations may be beneficial. It allows them to have sort of things happening in the big population of microbes that randomly might introduce some change that actually allows them to adapt to their environment better, and the microbe to which that lucky mutation happens will then survive better than its brothers and sisters. And natural selection will favor those that have a greater fitness. So for microbes, mutation is beneficial, and if it's not, those guys just die out. The population continues. Now, what kinds of changes would be acted upon by natural selection? Well, one way that you can have an impact is if you make a change in a regulatory region of DNA that might be affecting how genes are regulated, right? So remember the structure of the operon and that operator region and the promoter region, those non-coding regions that tell the microbe whether or not these genes should be made or not. You could mutate that region so that it changes how those genes are used. Or you could mutate the proteins themselves. And if you did that, you might change the ability of the protein to do something. A change in the DNA is referred to as a change in its genotype. And in bacteria and other prokaryotes, this is basically immediately going to have, if it has a consequence, it's going to immediately have a consequence because bacteria have only one copy. This is not true in eukaryotic cells like humans, right? So how do we know this is true? When you think about the genetics that you know, do you know of any traits that you can have silently pass on to your kids, but you yourself are not affected. Do you remember sort of hearing about that when we talk about genetics? Let's say something like eye color. There's a variety of genes that encode different eye colors. Some of those eye colors are dominant. So let's suppose you have a dominant eye color and your partner has a non-dominant eye color, then your kids are more likely to have the eye color that's dominant. It depends on details of genetics that we're not going to go into, but basically that's because we have 2 copies of everything. But prokaryotes don't. They have 1 copy. If there's going to be a change in something you can detect, then we call that the change in the phenotype. So, for instance, perhaps we mutate the gene for one of the genes in tryptophan biosynthesis, okay? And we disable that protein. Then tryptophan can't be made, and the only way that microbe's going to survive is if it can actually take tryptophan up from its environment, which is possible. But that means that that particular mutant is only going to survive if you supply tryptophan in the medium. Another kind of mutation is if, let's suppose you're a bacterium and some fungus nearby doesn't like you very much and makes an antibiotic to kill you. And that antibiotic, let's suppose it's penicillin, targets an enzyme that makes cross-links in peptidoglycan and you're gram-positive. And so you're sensitive to it. If you could somehow change the structure of your cell wall so that penicillin no longer was affecting you, you would become resistant, right? So there is theoretically a way that you could develop resistance to antibiotics by making some mutation in your genome that then changes your structure and that whatever is being targeted is not there anymore. There are 2 important things to say about this. This is not the most common way that antibiotic resistance develops in pathogens in humans. That is usually conferred by horizontal gene transmission, which we'll talk about. That's the first thing. And the second thing is that, I want to get to that in a second, so let's just skip ahead to get to that point. A couple of things about mutations, some terms to know. Point mutations are a change in 1 letter, so it's a single letter typo. A missense mutation means that whatever mistake you're making changes the amino acid. So for instance, here's the original sequence, right? These 3 letters make methionine, A and G, we know that. The next 3 letters make lysine, and then phenylalanine, and then glycine, and there's a stop codon. So here's our final protein. If we make a change to this 3-letter codon here, changing its code from glycine to serine, that is a missense mutation. A nonsense mutation means that in a place that's not normal, here's the normal place for a stop codon. If we make a change that introduces a stop codon sequence early, we call that a nonsense mutation. A frame shift mutation is where these 3-letter codes, which code for all the things, if we were to stick an extra base in here somewhere or take one away, we would shift the register that's being read, and then all of the later amino acids would be coded incorrectly. So frame shift is when you insert or delete 1 or 2 nucleotides or some multiplier of that. If you actually introduce something in multiples of 3s, you keep the frame. So how often does this happen? I already told you that in spontaneous mutation rate is about 1 in 10 to the 9 base pairs, or 1 in a million genes. Okay, 1 in a million genes, we have a mistake that's introduced by DNA polymerase. So that's pretty good. I mean, that's like, you know, if you had this error rate in your life, you'd probably be a superstar running several corporations. Mutagens increase the spontaneous error rate up to, 1 in every 1,000, or 1 in every 100,000, somewhere in that range. So it gracefully increases the rate. So what kinds of things are mutagens? There's 2 we have to consider: chemicals and radiation. So chemical mutagens are things like these chemicals shown here. For instance, some chemical mutagens are analogs. They look like one of the bases, but if your enzyme uses them, it actually has different base pairing, and so it changes the sequence of the DNA in that way during replication. Radiation has a number of possible consequences depending on what kind it is. So some radiation is really powerful and it chops the DNA, and then there's a break and it has to be repaired, and the repair process is mutagenic. But some radiation, like ultraviolet, actually instead causes different nucleotides to bind together. So here's ultraviolet radiation. This is common. Where does this come from in our world? In the sunshine. Okay. What do we know about UV that's good? That means it's not as damaging as it could be. It has low penetration. Okay, so what I'm about to tell you happens to your skin cells that are on the surface, right? UV can penetrate into the cells there and cause problems in the DNA of your skin cells, but it's not going to get into your liver. So that's good news. That's why you don't have UV-induced liver cancer, but you might get skin cancer. So UV radiation causes adjacent thymines to bind together, which creates this kind of lump that cannot be read by the DNA polymerase during replication and has to be chopped out and repaired. And the chopping out and the repairing is done by different enzymes that are not as good at copying, so they introduce mistakes when they fix the DNA. Okay? So how do we know if we have a mutant? How can we tell if a population that previously was sensitive to, say, an antibiotic now is resistant? How can we test chemicals to see what their effects might be? We have two ways of doing this: positive and negative selection. Positive selection is when we're looking for a mutant cell that's doing something it couldn't do before. Okay, so what we'll usually do is expose a population to something that will kill them and look for the guys that survive, and they have now mutated to be resistant to something. Negative selection is when we're looking for the other kind of mutation, a mutation that knocks out a function. So perhaps it could be something like normally this guy makes tryptophan, and we're looking for a mutation in one of the enzymes that makes tryptophan. So now we're going to look for things that don't grow. And this is tough, right? This is easy. We put down the thing that's going to kill them, then we put some cells on top, we incubate, we look for the 1 or 2 lone survivors. This is tough. This is, we put down something that normally the cells would survive on, we put the cells on top, and then somehow we have to just magically know that the ones there are dead. Like, how does that work? Okay, so we'll talk about how we do that. This is a process of replica plating. So what we do is we have whatever medium we're testing. Let's suppose that what we're looking for is the amino acid, some sort of nutrient that normally the organism is capable of making. That's the end product of a metabolic pathway. So we make 2 kinds of media: rich media that everyone can grow on, and media that only the guys that can do that metabolic pathway are going to grow on. But we're looking for mutants that won't grow on it. So on the rich media, we put down cells wide enough apart that they form individual colonies. Then we take some material like a sterilized velvet, and we set it down on these colonies after they've grown, and we use it like a rubber stamp to copy it to both the rich media and to the medium that is lacking some nutrient. We let both of those things grow. This one tells us which colonies were successfully picked up by the velvet. On the rich medium, everyone's going to grow. This one tells us which guys are missing. And then we go back to this spot and we go, okay, well, there's our mutant. Now, if we want to understand what's happening when we add chemicals and is there some mutation that's occurring as a result of the chemical, what we can do is we can culture things in the presence of the suspected mutagen, and we can use the process of mutation itself as a readout. So we can put the cells on something that doesn't have a nutrient that they need. And we can add this mutagen and see if things now grow that weren't growing before. This is a little bit hard to conceptualize, so let me say that again. We have, in the previous example, we isolated a guy because of a very small mutation, couldn't make some nutrient anymore. Okay? It was probably because of a very small mutation, like a change in one base pair, that changed an enzyme that's part of the metabolic pathway. So that's one change. That means that you could potentially change it back. So then we take that guy that's not able to make that nutrient because of one change in the DNA, and we add a mutagen that mutagenizes randomly, but there's some chance it's going to change that one base pair back to what it was before. And we look to see if that's happening and how often it happens. It could happen spontaneously. That's totally possible. So we have a control where we don't add the mutagen, and we see how often it normally happens on its own. And then we compare that to when we did add the mutagen to see if it's happening more often, and that is how we determine if something is mutagenic. Okay, so last but not least, we need to talk about these three terms, which have to do with when DNA is being shared between prokaryotes. So that means we have to understand these two terms, vertical transmission or vertical gene transfer, and horizontal transmission or horizontal gene transfer. Vertical gene transmission is just basically replication during cell division. That's simple as it gets, right? We're a parent cell. We make an extra copy of our DNA. We give it to our kids. That's the normal way that we pass genes down through the generations. So here we have an example of vertical transmission. Horizontal transmission is when you give your genes to your friends. Maybe they're not your friends. Maybe you don't like them so much. But you're taking your genome and giving it to your friends and neighbors and not to your kids. You're giving it to cells that are sort of essentially in the same generation as you are. This can happen between members of the same bacterial species. It can also happen between members of opposite or different species. And there are three natural mechanisms: transformation, conjugation, and transduction. So let's go through all of these and understand what they are. Transformation, I want to warn you, is one of these biology terms that has different meanings in different contexts. It's kind of like vector. You'll run into vector a little bit later in this course. And if you're in physics, vector has one meaning. If you're in biology, it has several, actually. And then it depends on what you're talking about as to what it means. So transformation is a term that's used sometimes in cancer biology to mean when a normal cell becomes cancerous, and we're not using it in that context. When we say it in microbiology, we mean naked DNA uptake by bacteria. So what the heck does that mean? Okay, imagine this. I'm an E. coli cell. I don't look like that at all because I'm a rod, but never mind. And I have my normal bacterial chromosome. Here's my friend, E. coli B, and we're hanging out together. And I have some genes on a plasmid, and we're going to talk about what that is in a second. But I have some extra genes. And I don't know, my time is up, I die. Okay, so sad. And when I fall apart, my genome is spilling out, and my plasmid goes out into this extracellular fluid between me and my friend. And at a very, very low rate, this plasmid or parts of this chromosome might end up getting into my friend here. Okay, so either part of this gets in, or maybe some of my DNA gets in. And whatever genes get in, let's suppose this was, I don't know, not going to be penicillin resistance because E. coli are already resistant to penicillin, something like tetracycline resistance. Suddenly my friend is also tetracycline resistant. So when we say naked DNA in solution, that is, in nature, a kind way of saying we got DNA from somebody who died. In the lab, we frequently do this. We kind of skip the dying step. We just have ways to generate whatever DNA we want. Maybe it's a plasmid, maybe it's whatever. So we just make some DNA, and we put it in some tube, right, with solution, and then we put some cells in. And that is also transformation, and we're going to do that in the lab. Okay, so transformation, naked DNA in solution is taken up by a bacteria. Conjugation. Conjugal. What is conjugal? Conjugal relations. Conjugal visits to the prison. What's conjugation? Bet you didn't know prokaryotes had sex, but they do. Conjugal, okay? So conjugation is when bacteria transfer DNA between each other, and usually this is between cells of the same species. And there's two different ways, depending on if you're gram-positive or gram-negative, and we'll talk about both of them. And then finally, transduction. Transduction is when the bacteria get their DNA by viral infection. Okay, so you can sort of think of this as an STD. That'd be one way to think about it, just if you want to remember it more easily. Although there's no sex involved, nonetheless, it is a viral infection that gives you some DNA. All right, so let's look at this. Overall, these mechanisms are called kinetic recombination, where there's a recipient cell, and here's all its normal genes. And then there's some other genes that get into the cell by some method, okay? And then eventually end up being incorporated into the genome. So first, let's talk about transformation. Historically, transformation was first described and understood as a result of this interesting experiment. So this is dealing with pneumonia, and several times I've mentioned now that pneumonia is caused, some form of pneumonia is caused by bacteria. And you'll also possibly remember that I've talked a lot about how that particular species is virulent because of its capsule, which protects it from our immune response. And that there are very closely related species of that bacterium that don't have a capsule and are not pathogens in humans, okay? So in the process of understanding this disease biology, this experiment was done, it had some very surprising results. So here's a mouse, and if you take and it also is, just like us, is susceptible to this pneumonia bacteria. So if we take a live bacteria with capsules, so the pathogenic version that's alive, and inject that into a mouse, the mouse dies. I know, it's a very sad experiment. It already starts out very sad, so just bear with me. So then if this pathogenic bacteria multiplies in the blood of the rat, and if you take a sample and grow it on a Petri dish, you'll see colonies that are, and when you do an analysis of these colonies, you can see that they are basically what you put into the mouse. On the other hand, if you take this mouse and inject it with this sibling species, this very similar species that just doesn't have the ability to make a capsule, because it doesn't have the genes to make the enzymes to make the capsule, then this guy does not die. This does still replicate a little bit, and when you isolate the samples from the mouse, you get back what you put into it. Interestingly, if you take this pathogen and you heat kill it first, then you can inject the mouse and you don't get anything out because those heat-killed pathogenic bacteria don't multiply at all. The mouse is totally fine, okay? But if you take these two things together that normally don't do anything, and you inject them at the same time into the mouse is dead. What happened? What you're basically putting into the mouse is the pneumonia bacteria A, which has a capsule and is pathogenic, but it's dead, which means that it's degrading and its DNA is leaking out, and the DNA includes genes for making the capsule. And alive, it's related species that has no capsule and is normally harmless, but it's sitting, in the same environment, and all these genes are around, and some of the time, the genes for making the capsule are going to get into this guy, and suddenly he's going to turn into the pathogenic version. Okay, bacterial conjugation. So there's two forms of bacterial sex. Gram-negatives do it with pilus. So what they do is they have genes that make this special pilus. And you remember, the pilus is Normal pilices are just these spikes sticking out that are have certain functions and they're kind of smallish. But there's this specialized pilus that can be encoded by a gene, genes on a plasmid, we'll talk about that later on, that makes a longer tube, and it can connect with another cell that doesn't have this and actually make a hole, and then what happens is that the chromosome is replicated and sent through this tube and invades the other cell. And it takes some of the DNA from this host, or from the donor, into the recipient. So this is what happens in some gram-negatives. For instance, E. coli can do this. Gram-positives don't have this ability. And the difference is, we don't have to go into detail, but you can imagine there would be differences because we know something about the differences between these types of cells, right? The cell wall is different. So whatever the pilus is doing is probably not going to work on this different structured cell wall. Instead, some gram-positives have the ability, if they're really close to each other, they can make actually a mating bridge, which is just a tube between the two cells. And again, DNA from the donor can go into the other cell. And so it's a similar end result, which is that if there's genes in here that make proteins that this guy doesn't have, he may end up with those new features, those new abilities. So this is just a schematic diagramming. Whether you're using a mating bridge or the pilus, you're basically transferring DNA into a recipient. Okay. Plasmids we'll come back to in another lab in a couple of weeks. It's going to take us about 2 weeks to get there. But plasmids are like mini chromosomes. So just for a sense of scale, let's look at the difference in size. Here is some prokaryotic cell. And here is its genome. This is its normal chromosome. And let's suppose there's like 1,000 genes on it. Over here we have a plasmid. And in terms of structure, the plasmid is identical in that this is just a DNA sequence that's circular, and a plasmid is just a circular piece of DNA. But what is different? How many genes is on this plasmid? If this is 1,000, how many genes do you suppose this is? Your typical plasmid is only going to have 3 or 4 genes on it. Okay, so what do plasmids carry? Well, they're probably not carrying really absolutely vital things. Okay, and one of the reasons for that is that the one major difference besides size is that this bacterial chromosome has an origin of replication, and also a way to be faithfully inherited every time you have cell division. But plasmids, while they are capable of being replicated, are not segregated in the same way. So if you have plasmids, when you go through cell division, there's, either only one daughter is going to get the plasmid, or even if there's several copies of the plasmid because they were replicated during the cell division process, they might all just by coincidence be on one side of the cell. So there's no segregation machinery the way that there is for chromosomes. So what you're not going to have on plasmids, you're not going to store things on them that you absolutely need to survive. Like, you're not going to put your tryptophan operon on a plasmid. That would be dumb. But the plasmid might have something like penicillin resistance on it, a gene that encodes the ability to resist penicillin. And so then if you have this plasmid, you're going to be able to resist penicillin. If you go through cell division, some of your daughter cells might lose it, and now they die if you give them penicillin. Other ones might still have it. Also, the plasmid is the most likely type of DNA to be transferred during a transformation event. So here's our plasmid. It's a circular piece of DNA. It has an origin of replication, a handful of genes, and it often will have things on it that are just enzymes that give you extra abilities, but that you wouldn't necessarily need all the time. Or resistance factors, for instance, antibiotic resistance might be found on plasmids. Okay, the last thing we need to talk about is transduction. So this is the getting DNA by having a viral infection. So the type of virus that infects bacteria is called bacteriophage. That is the name for all viruses that infect bacteria. Here is our bacterial cell getting infected by a virus. The virus has some DNA in it, and I don't know that if we've talked about this or not. I do know that we've talked about the fact that viruses have either DNA or RNA, right? But bacteriophage all have only DNA. Other animal viruses and plant viruses, it varies, but bacteriophage are always DNA. So here is a bacterial infection, and what happens in a bacteriophage infection is that the DNA gets injected into the cell. It starts to cause a lot of mayhem, including breaking up the chromosome of the infected bacteria. In the process of making more viral particles and more genomes, some of this broken up bacterial DNA could accidentally get packaged into a viral capsid. And when this viral capsid goes and actually injects its DNA into another recipient cell, what it's injecting might not be a viral genome. It might actually be DNA from this guy, which is now in this recipient. So that is transduction by virus. Okay, so to close, we're going to go back to this, which you may or may not recall we talked about in the very beginning of this chapter. So remember that there is a strain of E. coli which, unlike our normal harmless gut bacteria, is pathogenic. And I also mentioned that it's called O157H7. It's the one that has been associated with various food poisoning outbreaks. And it's pathogenic because it makes this toxin that most E. coli don't make. It's a Shiga toxin. Shiga toxin is made by Shigella, a completely different species. So how did this Shigella toxin end up in E. coli? Did it just mutate into existence? That seems so unlikely. Okay, so where did it come from? It came from horizontal gene transmission of some kind. Now, it probably didn't come from conjugation because Shigella is a different species, so it's probably not from a mating bridge or from a pilus. It could theoretically have come by transformation, right? So the Shiga toxin might have been something that was on a plasmid, and the Shigella and an E. coli were hanging out together at a coffee shop, and the Shigella suddenly dropped dead. His DNA was lying around. The E. coli got it. Okay. The other way it could have happened is if both species could be infected by the same type of bacteriophage. So bacteriophage A infects the Shigella. The Shigella chromosome breaks down, gets packaged into new virus particles, which go and infect E. coli. Now E. coli has this new gene that it didn't have before. So in this particular case, that latter scenario is what happened. It was a viral infection. And so basically, the harmless E. coli got infected by a bacteriophage that was carrying a Shiga toxin gene that got incorporated into the E. coli chromosome. What does Shiga toxin actually do? It inhibits protein synthesis. It's similar to ricin, if you've heard of ricin. That was used in bioterrorism in Japan. So basically what happens is that this toxin enters the cell and it chops a little piece off of the small subunit of the ribosome, and it then basically stops protein synthesis. Okay, so we're going to switch gears entirely and